Hi guys, so I am ready to introduce you to the star of our laboratory experiment. This is MGH. So this is the protein that we are going to be working with throughout the course of this entire laboratory course. Again, we've had to rearrange things just a little bit here. So we're front loading some of the wet lab experiments. So I wanted to introduce you to who we're going to be working with and let you know kind of the next few weeks how they're going to be laid out in terms of labs. So this is MGH. And MGH is an acronym that stands for the 3 domains of the protein that we're going to be studying. So I do want you to know what this acronym stands for and what each of these players is doing and why we're studying this protein. So the M stands for malate dehydrogenase. So malate dehydrogenase is an enzyme. We're interested in actually looking at its enzyme activity towards the end of the course here. So that is the protein of interest to study. Now the challenge oftentimes with studying proteins or purifying them is they may not be visible. So what we do is we actually attach a green backpack, if you will, to this protein. So we make what's called a fusion protein. So we actually have 2 proteins that are fused together. So just to highlight here, this isn't that we make these 2 proteins separately and we fuse them together. We actually have a gene sequence that has these 2 proteins immediately adjacent to one another, so that when they are synthesized, they're synthesized as one unit. But they will fold independently into their separate domains. So we've got our malate dehydrogenase, which is really our protein of interest. And then we have a green fluorescent protein domain. So this folds independently and separately, but what this allows is for our protein to be neon green in color. We saw a little sample of this protein when we did our first wet lab, when we kind of worked with some basic biochemistry techniques. Now the H stands for a histidine tag. So this is 6 histidines that are in a row at the very end of our proteins. Remember, proteins run from N terminus to C terminus. So this is at the C terminus. This is 6 histidines in a row. So 2 things are important about that. The first is that you're not likely to find 6 histidines in a row in nature. So that makes something very unique for our protein. And the second thing is we're actually going to be able to use that protein as a way to go molecular fishing for our protein. So just as a little preview, when we make this protein, it is going to be in a slew of thousands of other proteins. So we want to isolate this protein from everything else that we don't want. That's a big theme that we're going to have in the course here. How do I isolate and separate what I want from what I don't want? And this histidine tag is going to allow for us to do that. We are going to use column chromatography to purify this protein, and we're going to use a column that binds specifically to consecutive histidine residues. So again, most naturally occurring proteins are not going to have that, so they won't stick to the column, but our protein will. So this histidine tag is going to help with purification. The green fluorescent protein is going to help with visualization. And this malate dehydrogenase is really the catalytic enzyme that we're interested in studying. So that's an introduction to the player. So let's get into some details about what labs we're going to have that are involved with this player. I first want to kind of tell a little story about how proteins are made. And usually this is kind of story time in lab, but I'm going to do kind of story time right now just virtually here for you guys. So the first thing I kind of want to highlight is that when we think about proteins that have been studied historically, the proteins that were studied very early on were ones that were easily isolated from biological tissues. So these easily studied proteins at this point in the course, we would have already kind of talked about these proteins in lecture, but we're getting a little bit front loaded here with our wet lab work. So 3 proteins that we are going to study in lecture, so we're just going to introduce them now, are hemoglobin, which we abbreviate HB, myoglobin, which we abbreviate MB, and insulin. And I've got these grouped here in this way because hemoglobin and myoglobin are oxygen-carrying proteins, but hemoglobin is found in the blood and myoglobin is found in tissues. And so hemoglobin and insulin are both blood proteins. And so one of the things that we'll kind of notice here is that they're from places that were easy to obtain from usually animal sources. So whether it was animal tissue that you could easily isolate myoglobin from, actually myoglobin was first isolated from sperm whale because you could have so much muscle tissue there. And then all you needed to do to isolate hemoglobin and insulin, which are very water-soluble proteins, was take a little blood from an animal, and you'd be able to isolate that. So that's why those were proteins that were well studied early on. Just kind of thinking about some other proteins that were well studied early on, and this has to do with sort of their therapeutic benefit. So I'm going to call these therapeutic proteins. Obviously, insulin is another very important therapeutic protein, and also growth hormone. So early on, one of the things that happened is maybe we could use another animal isolate. Actually, our case study on insulin will delve a little bit deeper into this, talking about how there might be some animal sources of insulin that would be compatible for use in humans, and so that could be a viable way to get insulin to be able to use for humans as a therapy, right? Because clearly if we have a human that needs their own insulin, they can't really provide it for a patient who can't produce and thus needs insulin. And so we may have animal sources for that. But growth hormone presented a real challenge because growth hormone is very human specific. Humans need human growth hormone. And usually I'll throw this out to the group and I'll say, does anyone know where we get human growth hormone? Human growth hormone is produced in the pituitary gland, so in the brain. So the only place to get human growth hormone is from cadavers. So this presented a challenge, especially as we started to identify diseases, prion diseases, that were communicated via brain tissue. And so all of a sudden it became problematic to use cadaver tissue to isolate human growth hormone. So we were really at a loss then of developing technologies to develop and to make these therapeutic proteins. So along comes the 1980s, and along comes a field called recombinant gene technology. And when I was in college in the early 90s, this was all the rage. Everybody wanted to be an RGT major because that was like the up and coming field. And really what this involved is using bacteria essentially as factories to make proteins. So we're going to use bacteria as factories to make proteins. And so that's the technology that we're still going to be using in today's lab to be able to make a protein of interest. So let me just describe how it works. It might be something that you've learned about in another course. Basically, you have a DNA plasmid, which is a circular piece of double-stranded DNA that has a few bells and whistles with it, but you have the ability to insert your target gene into this plasmid. So you have your DNA sequence that can be made into your desired target protein. So what we do is we go through a 3-step process here. Transformation means taking this target gene and putting it into a bacterial cell. So that might be the first T word here that you didn't hear or haven't heard of yet. So transformation is basically going to take, and we're going to put the plasmid into a bacteria cell. Now, once that plasmid is in the bacteria actually don't really know how to differentiate that DNA sequence from their own circular genome. So what happens is they just use it and make the proteins off of it. So it's almost like we're hijacking their machinery to do our work. And the work that happens with any DNA sequence that's made into protein is that DNA is transcribed into messenger RNA and then translated into protein. So that probably is something that you've heard of before. So this allows us to take a target gene and then make a target protein in bacterial cells. So this sort of revolutionized the ability to make not only human insulin, but human growth hormone. And so it just really took off. It became a very significant field, and now there's still a lot. If you have insulin that you have to take, if you actually look at the bottle, it probably says recombinant insulin. And that's because it's made through this process. So we're going to use this same process, and our target gene is actually going to be a gene that's going to make malate dehydrogenase immediately adjacent to green fluorescent protein that has a C-terminal histidine tag at the end. So that's the protein that we're going to make. That's sort of the technology that we'll use to do this. This process is called protein overexpression. The process of using bacteria to transcribe and translate our own plasmid DNA that we put in is called protein overexpression. So again, a little bit of just summary here. Some things I've already said. What is a plasmid? It's a circular double-stranded piece of DNA. We can artificially then carry genetic information into another host cell. What's really neat about so much that's happened in molecular biology is these were naturally occurring processes that we've sort of hijacked for our own benefit. So plasmids are how bacteria sort of swap and exchange information. We've just sort of utilized that to say, you know what, let's put our information on that. Let's put our target gene in there. So this process of protein overexpression means that we are going to transform or put our plasmid into another cell, and then we're going to transcribe and translate that into our desired protein. And so for us, it's going to be MGH. A little bit of plasmid anatomy, which is going to be important as we move into our first set of labs, which are on DNA work. Plasmids contain what we call a multiple cloning site, or an MCS. This contains unique stretches of sequence that are going to be recognized by what we call restriction enzymes. I'm going to call them molecular scissors. That's going to allow us to snip in and snip out the pieces of DNA that we want. And so that multi-cloning site is just one of those bells and whistles that we have on plasmids. An important bell and whistle that we have is what we call a selectable marker, and it's labeled here as an AB for antibiotic resistance. So one of the special pieces that will be in our plasmid is the ability to make a protein that will allow it to survive in the presence of an antibiotic. And this is important because, again, in our theme of how do I separate what I want from what I don't want, it's going to allow us to separate and isolate cells that have our plasmid, which we want to grow, from bacteria that maybe don't have a plasmid at all or have a different plasmid that has a different selectable marker in it. So it's going to allow us to do that selection. I'm not going to mention too much about this origin of replication, but that is another integral part of a plasmid that allows it to do its job. So the two things that we're going to focus on are that multi-cloning site and then the selectable marker. Sometimes this is called an antibiotic resistance gene. A couple of things just to highlight here. So why are we, again, working with bacteria? Two times that we're going to work with bacteria, or two sort of themes of things that we'll be doing with bacteria. We're going to be doing work with plasmid DNA, and then we're going to be doing work with protein. So highlighting here the first two labs that we'll have is plasmid DNA work. We can use bacteria to amplify, store, manipulate, isolate, purify plasmid DNA. Now, one of the things that's key that I want you to understand is there's two different strains of E. coli that their own bells and whistles as strains of E. coli allow them to perform different tasks in the lab. The strain that's called DH5 alpha is going to be the strain that we're going to use when we're doing things like storing, manipulating, isolating, purifying plasmid DNA. And that's because it's going to make a lot of copies of that DNA plasmid, which is going to be what we want if we're doing some plasmid manipulation work. If we want to store our plasmid for long periods of time, we're going to have it in a DH5 alpha cell line. Now we're going to look at the flip side. When we're starting to do protein work and we want to overexpress a protein, we need to have a special cell line that's capable of doing that protein expression work, of taking that plasmid and then transcribing and translating that gene that's on there. That different cell line is called BL21DE3. So I know it sounds a little bit like an alphabet soup. Those are two things that I want you to know. DH5 alpha is the cell line that we use for storing, manipulating, purifying plasmid DNA. When we want to do protein overexpression, we have to have it in a different cell line, which we call BL21DE3. Okay, so what we're going to do for the next few labs is we're going to have two labs that we're going to have on plasmid DNA work. The first lab is going to be isolating plasmid DNA from our storage line. The lab we do after that is then putting that plasmid into our overexpression line. So those are going to be two DNA labs that we'll have. And then we're actually going to have 2 protein labs. The one that's listed here first is that we are actually going to then use that BL21DE3 protein overexpression cell line, and we are going to make our MGH protein. Now, after we've made that MGH protein and purified it, one other lab that we're going to do is a kinetics lab. We're going to focus on using a different protein that we know behaves really well to learn the techniques of kinetics, but then we're going to try and see if we can get this malate dehydrogenase enzyme. We're going to see if we can get this enzyme to provide some reliable kinetic data. All right, so that's why we culture bacteria. 2 different things. We can do some DNA work, we can do some protein work. I want to talk briefly about how we culture bacteria and 2 different sort of systems that we might use. You'll notice that a lot of things that we do here, it's going to be sort of this binary, this versus that. I think it's a good way to sort of compartmentalize information here. So you can grow bacteria on solid media, so that's growing them on like bacterial agar plates. Well, why would we want to grow bacteria on plates? We can then separate bacteria into individual colonies. We know colonies grew from a single cell, so we represent a clonal population. So every one of those bacteria are identical because they originated from a single starting cell. So when we want to work with one specific isolate, we're going to spread colonies on a plate, and that'll allow us to pick a colony from a plate. And when you're doing that, you know that you are essentially picking what equates to one single cell that has grown to a significant volume where you can actually see it. Okay, we can't grow a lot of bacteria that way, but that's not why we're growing it on that solid support. We're growing it on the solid support so we can separate things. And that's different from growing bacteria in liquid media, or what we call broth. Now, when we grow things in broth, we can have a small kind of a sample of broth and grow a small kind of bacterial culture. Or we can grow up to, several liters worth of bacterial culture. And so depending on how many cells you need, what your downstream application is, you can have different amounts of cells that you'll grow. And again, sometimes we might be growing them in broth because we're going to be isolating plasmid DNA. That's what our first lab is. That's going to be growing E. coli as DH5 alpha. But when we're overexpressing protein, we're also going to be using this liquid media, but that will be the same plasmid, but in a different cell line, BL21DE3. So this is, again, what it looks like when we grow bacteria in a culture. Now, if you see a cloudy culture, that means there's bacteria growing in it. So these cultures, this broth, when we first start it, it's nice and clear. It doesn't smell bad. But when bacteria grow in it, it gets cloudy, and then it doesn't smell so good. All right, so I want to take the last little bit here and just give you the 30,000 foot view of what we're going to be doing over the rest of the course with our wet labs. So what we're going to be doing right now is front loading ourselves so that we get all of our wet lab experiments done, and then we'll be able to talk about and analyze the data and do all of that sort of if we need to be virtual later on, or it'll just be, we'll be able to have a dry lab where we kind of analyze our data. So 2 experiments that we have that are DNA based. These are the first 2 that we're going to do. So the first one is this week coming up, so it's September 10th, and then September 8th right now is what we're going to have planned for the kind of second part of this experiment. So the first part is going to be DNA plasmid mini prep. So I'm going to run through just kind of in a very global sense sort of what that is. And then the second week's lab is going to be transformation and double digest. Okay, so we've got our bacteria here, and they're going to have our plasmid inside. So when we're doing DNA work, remember, we're going to have DH5 alpha bacterial cells, and we've got our plasmid that's inside. Now, notice I only drew one little circle inside. That's not to say that there's only one plasmid inside, but just for kind of simplification here, I'm just going to say that there's only one plasmid inside. What we're going to do here in our plasmid mini prep experiment is we need to lyse the cells open. So we're going to use that term a lot. Cell lysis literally means breaking apart the cell, and we literally spill out the contents. Remember, when we have a eukaryotic cell that has all of our organelles like we have, we're going to have like compartmentalized things that come out. When we have a prokaryotic cell, like a bacteria, we literally dump everything out and it's just all out there on the floor. So when we lyse this open, we generate what we call cleared lysate. So this is a term that we're going to see in several labs here. This is a term that when you break open cells and you remove the soluble part from the insoluble part, that soluble part is what we call cleared lysate. Okay, so cleared lysate in this case is going to have a whole bunch of stuff. When we have on our next video kind of what that stuff is, we'll talk about all the different things that are in there. But what we want to do is we want to isolate our DNA plasmid from everything else that's there. Remember a theme that we have. How do I isolate what I want, my DNA plasmid, from everything else that's there? Essentially the rest of the factory that made or was housing that plasmid for us. So what we use to do that is a technique called a plasmid mini prep. And I'm not going to go through the details for how this works, but basically at the end, we're going to have our plasmid that we're going to have in a tube with some water in there. And so we've isolated our plasmid from everything else. And so what we're going to do with that plasmid is we want to quantitate it. We want to find out what's the concentration of that plasmid. So we're going to use spectrophotometry to do that. Okay. The other thing that we want to do is we want to send it out for sequencing. And the reason that we want to do that is if we were to spend the time, energy, effort, resources to overexpress a protein from a plasmid, we'd like to make sure that the gene is the right sequence, that there wasn't a mutation that happened, that we didn't accidentally grab the wrong stock of cells so that it's the wrong plasmid that's in there. So it actually only costs about $5 to do sequencing. We send it out to a facility that does it, so it's very cost effective. But before we move on and do any further work with it, we kind of want to make sure that it's the right sequence. So that's going to be what we do in our first week. I am going to have you guys watch another video to go through all of the background on how a plasmid mini prep works, so that when we come in, we basically have 90 minutes that we can get in, get out, and do the work that we need to do for a plasmid mini prep. Now, I will tell you, a plasmid mini prep you honestly can do in about 15-20 minutes, so it really doesn't take that long, but we want to make sure that we take the time to understand all the theory behind it first so that we're ready to come in and do that. All right, so that's our lab this week. So, next week's lab is actually going to be taking that plasmid that we quantitated it, and we're going to be doing something downstream. So that's sort of how this course is set up. Each of these labs sort of builds on something that we made or prepared in the week before. So now what we're going to do is we're going to take this plasmid and we want to add it to BL21 DE3 cells. So that process of transformation, we want to take and put this bacterial plasmid inside, and again, that is the process of transformation. So we'll talk about and we'll actually set up and do a transformation next week in lab. And then the other thing that we're going to want to do, so this is kind of the first thing that we're going to do. The second thing that we're going to want to do is another way to sort of check and make sure that the plasmid is what we think it is. We're going to do what's called a restriction enzyme digestion. Okay, it's also sometimes referred to as a double digest. So what we have here is we're basically going to have our plasmid, and we're going to use what essentially equates to molecular scissors to cut. Remember I told you that there's that multi-cloning site? We're going to use the scissors that match up with that sequence that we have in that multi-cloning site. We're going to have 2 different kinds of scissors, and what they're going to do is they're going to digest or break apart this plasmid, and we're going to be left with that piece and then this piece. And then what we're going to do is we're going to run these on an agarose gel. So we're going to do gel electrophoresis. And in doing gel electrophoresis, we're going to be able to see whether or not we made the right size pieces for doing our double digest. So this is, again, another way to confirm that we have the correct plasmid. So just introducing you to a bunch of different DNA techniques that you might need to use in a biochemistry lab. So 2 labs for our DNA techniques. And then we're going to have 2 labs that we do protein work with. So again, highlighting what those different experiments will be. Sort of behind the scenes, what I'm going to be doing is taking, once we get, let me highlight here, once we put this in here, and then we're going to have our plasmid in Bl21DE3. Okay, what we're going to have here is work that I will do sort of behind the scenes. It's not anything super, technologically advanced. It's just a lot of babysitting of bacteria to get these bacteria to make the protein. I'll explain everything that we're doing here, but it's not really worth your time and effort and energy to try to do it yourself. We're just going to get to the end product. So behind the scenes, I'm going to take these bacteria, I'm going to get them to make the MGH protein, I'm going to get them to spill their contents out, and we're basically going to be starting with a cleared lysate. So highlighting again, we had Bl21DE3. And to give you the quick overview of what happens here, I'm going to be growing these bacteria, I'm going to be babysitting them, monitoring their growth, making sure that they're not getting too dense and they're growing well enough, that they're happy, they have enough food, all that kind of stuff. We're going to add a chemical called IPTG. So we're going to add this chemical, and that's going to be a signal that tells this bacterial factory to make my protein. Okay, so this is going to be overexpress our MGH protein. Then what's going to happen is I will lyse the cells open, and we will get this cleared lysate. Let me actually do this. I know, sorry guys, you can't do this. So we get our cleared lysate. So this is stuff that I'm going to be doing behind the scenes. I'm going to take those bacteria, the Bl21DE3 cell line that has the plasmid, I'm going to add a special chemical signal that gets them to turn into the factories and make our protein. Then I'm going to lyse open those factories and get out the mix of everything that's there, that cleared lysate. Just highlighting the difference here, we did have a cleared lysate here as well, but what we wanted to isolate from that was DNA plasmid. DNA plasmid will still be here, but now what we want is we want to isolate MGH protein. And so we're going to use different techniques because we need to isolate something different. So we're going to isolate that MGH protein. Just very superficially, what we're going to have here is we're going to have a column, and it's going to have a resin. And that resin is going to bind that his tag. So remember, we had our MGH, that H stands for that histidine tag. This resin is actually going to bind that histidine tag. Everything else is going to be able to flow through, and then we'll be able to isolate just our protein. Again, big theme is how do I isolate what I want from what I don't want. So we're going to talk about how the methods work for doing that purification, and then we're going to talk about how we analyze it. We're going to quantitate our protein. We're going to try to figure out which fractions, because when you do column chromatography, you isolate fractions. So we're going to have to figure out which fractions do we want to pool together, all that kind of fun stuff. So that'll be our first protein experiment that we do. The second protein experiment that we're going to do is a kinetics experiment. Okay, so very superficially here, if you've got an enzyme that reacts with a substrate, it can convert that substrate into product, and it's an enzymatic reaction, a catalytic reaction, because we regenerate the enzyme that can go and then repeat that process. So what we have here is, if we have a product that's colored, what we can do is monitor the course of this reaction. And we haven't really done any data analysis yet, but we've talked about what a time course is. If you measure, for example, the concentration of product as a colored species as a function of time, you should see that it increases. The slope of this line is going to be the rate of that concentration of product increasing, and that is related to the rate of that enzymatic reaction. There's a few other things that we have to think about doing, but we will do an experiment like this with an enzyme to be able to determine some kinetics parameters. So you'll have experience not only gathering the kinetics data, but then we'll spend a good bit of time in how we analyze that kinetics data. So just to highlight here what this enzyme might be, okay? This enzyme is likely to be something called LDH, which is lactate dehydrogenase. Okay, but we just made a protein that has malate dehydrogenase with it. So sometimes, based on how well our purification scheme worked, depends on whether or not we actually can get good kinetics data from the protein we made ourselves. So I always have one group that will actually try that. They'll go through the same techniques and everything to gather the kinetics data, but we'll do most of the analysis on our lactate dehydrogenase, because that's a commercial enzyme that I buy. I know it will work, so that we can get some good data from that. So that MDH is, again, malate dehydrogenase. And that M is part of the M in MGH. So we'll be able to take the protein that we've spent 4 weeks up to that point kind of making for ourselves, and we'll actually be able to analyze it. So those are the experiments that we're going to have for the rest of the course here in terms of wet lab experiments. There are several sort of theoretical or dry lab experiments that are sort of sprinkled throughout, or that we haven't yet done. So as time allows or necessitates, we'll kind of start working on those experiments as well. So I know this is a little bit different in terms of the normal sort of sequence of events that we have here, but I think we're going to be able to get in all of our wet labs, which I'm really, really excited about.